and the gene that encodes for NADH dehydrogenase, which is also, as we've talked about it, it's the first complex in the electron transport chain. So, a couple of questions to get us started. Describe the function. And then part 2, evaluate whether the energy yield will be affected. Let's not answer this just yet. But what I want you to do is first maybe take a minute and write down for yourself, what kind of things do you need to address to be able to answer this question? Particularly the second part. Evaluate whether bacterium's energy yield will be affected. So given the context of the problem, what do you need to make sure you address in responding to this prompt? Okay? Take a minute to get those thoughts together. All right, minute's up. What do we need, or I guess not what do we need, how would you address this? What are the things that you might want to focus on in responding to this prompt?
You could talk about like the purpose and function of dehydrogenase.
Okay, that's definitely one that's got to be addressed. So function of, I'll just label it DH right now, of the dehydrogenase. That's going to be important. What else? Alexi.
How is energy yielded?
Okay, and basically what that means is how is it produced, right? Could we be more specific with that, and particularly in what process?
The electron transport chain.
Yeah, in an oxidative phosphorylation. Good. So you probably need to know something about the ETC. Need to know, something about oxidative phosphorylation. What else? Those are probably the big ones. But I feel like there's something missing. I think we can get even more detailed than just this. What is the link between electron transport chain and production of ATP? It's not the electrons that are producing the ATP. Well, there's an electron acceptor. That actually is something that might become important. It may not. But whatever that final electron acceptor is, what's actually producing the energy? Or what's producing the ATP? I'm hearing some people starting to say hydrogen ion concentration. So we might want to address the hydrogen ions across the membrane. What's the actual enzyme that produces the ATP here? It's not a kinase in this case. The ATP synthase would be the last one. These are probably all things that you might consider bringing up. Some might be more important than others. Okay? So one of the things that I usually try to tell people is when they're approaching a problem like this and they go, I don't know where to begin. Start with what you know. This question really is asking you a couple of things, but these questions are hidden. One is, how does the electron transport chain work? And then the second part is, what happens when it's blocked? That's what evaluate means here. Okay? So to really answer this question, these are the things that we need to be able to identify. The function of a dehydrogenase, how energy is produced, particularly through oxidative phosphorylation. What is the connection between the movement of electrons and hydrogen ions and ATP synthase? Those are the things that we need to address. So here's what I'd like you to do. In the next, I'll give you, let's say 4 minutes this time. Write down for yourself a response. How does the electron transport chain work? All right, and then we'll pause and we'll talk about that, and then we'll move on to the next part of this. And be as detailed as you can. Give me some descriptions. How does the electron transport chain and oxidative phosphorylation work? Where do the electrons start? What's that? Complex 1. And where do they come from? NADH, good. So electrons come from, high energy electrons come from NADH, and they go into complex 1. Where do they go after that? They're not used directly by the ATP synthase. You're right that the NAD is oxidized, or the NADH is oxidized to NAD+, though. That's absolutely correct. What happens to these electrons? Think of the name. Electron transport. They're transported. They're transferred from one complex to the next. And where is this all occurring? That's something that we probably should know, too. Where is this process occurring? Good. Okay. Don't say mitochondria. That loses fingers. All right. So to get these electrons moving from one complex to the next, I'm not concerned with you guys knowing how many complexes there are necessarily. That's not the important point. The theme of electrons moving is important. And as they're moving, what's happening to those electrons? Are they gaining or losing energy? They're losing it, and how is that energy being converted, or what's being done with it? So what's being done with that energy that's being released by the electrons? Taylor?
Is it helping the hydrogen ions move?
Bingo. Exactly. Active transport. So those hydrogens are moving from inside of the cell to outside. Across these various complexes as those electrons are releasing energy. Okay. Ultimately, those electrons have to end up on what? Where do they end up at the end of this chain? They have to come off the chain. So what pulls them out of the chain at the end? If we're talking aerobic respiration, what's the final electron acceptor? That's the other term. Oxygen. That's what we're talking about, is the final electron acceptor has to pull them out of this chain. Oxygen is one option. If it's anaerobic respiration, it's something else. Nitrate, sulfate, carbonate are examples. So here's our O2. O2 gets consumed. H2O gets produced. And those electrons are now out of that chain so that that chain can continue to occur. And then what happens to those hydrogen ions? Move back through the ATP synthase or the ATPase, exactly. So that's this little thing here. And as they're moving back through, ADP and phosphate form ATP. And those hydrogens are, some of them, are being used for this process here to form the water. Okay. So that's the general process. That's the connection. Now, the question asks us to evaluate what happens to this if NADH dehydrogenase, or complex 1, is blocked, has a nonsense mutation that now makes it non-functional. Emma?
Since you're oxidizing the NADH in the
Okay. Absolutely.
I don't know how to explain it.
No, so basically what you would do here is to evaluate is you would argue, you've described what's happened, right? And now you think of, and you've described it in kind of steps here. The electrons come off of NADH to NAD. The electrons move from one complex to the next. That pumps the hydrogen ions across, and then the ATP synthase uses those hydrogens to generate energy. What you've just said is without this enzyme, a functional version, NADH can't be oxidized into NAD. In other words, the electrons can't be captured from it and used for the electron transport chain. If complex 1 is non-functional, do we get electrons moving to complex 2 or 3 or so forth? So the electron transport chain stops. What does that do to hydrogen ion concentration across the membrane?
It's higher than outside.
Well, it is originally, right? It's higher outside than inside. I guess maybe a different way to ask this question would be, are the hydrogens still being pumped across the membrane? No, because why? What does it require? It requires the electrons for that process to take place. And if hydrogen's not being pumped across the membrane, what's that do to the energy yield? Good. It decreases the energy yield. Okay? So what we've done here is we've walked through first a description of the process and used that knowledge to help us figure out what's going to happen if one part of this stops. Okay? One of the things that we haven't covered, and I just want to point this out more than anything else, we oftentimes say that Krebs cycle and the electron transport chain oxidative phosphorylation, they're mechanistically linked. In other words, if Krebs cycle shuts down, so does the electron transport chain. Because Krebs cycle is providing the NADH for, and the FADH too, for the electron transport chain to function. If this shuts down, you can't get oxidation of NAD. And in that case then, there's no available NAD to allow for the Krebs cycle to occur. And so if this shuts down, the previous step has to shut down as well. How would the cell respond? If the electron transport chain isn't functioning, how might it change metabolism? Yeah, fermentation is probably going to be a preferred method at that point, if it can perform fermentation, if it can perform it. All right, I'm going to go back to the get out of this real quick. Okay, I'm going to go back here. So then the other part of this asks, how might the mutation affect the pH across the membrane? It's a slightly different question than the one I asked about hydrogen ion concentration, but it's similar. And then how might this affect oxygen consumption? Here's what I want you to do. I want you to draw two graphs for yourself now. Normally, what would a cell, let's say that there's a limited amount of oxygen in a system and a cell is performing aerobic respiration. What would happen to the amount of oxygen that is present as an organism is undergoing aerobic respiration? And then show how would that change if electron transport chain is stopped. Okay? And then do the same thing for hydrogen ion concentration. And what this is requiring you to think about is how are those involved in this process as well. Try to work on this one with somebody else. This can be a bit of a tricky one, but I'll give you a setup of what this graph is roughly going to look like. There's the start. Your y-axis can be percent O2. We'll set it to 100% to start, and so consumption represents a decrease. And we'll just say that this bottom is time. Time of the respiration. So what's the general trend going to look like? I'll give you like a minute or two to think about this and the pH one. All right. So we know how the electron transport chain functions. Here's the membrane. Electrons move, bless you. Here's our complexes. And where are the hydrogens going? Outside the cell. If you can do this, or if you can kind of visualize this, this is a great first step. What's the problem say? What's the function of these isohumulones? Good. Makes the membrane leaky, right? In other words, how would we model that, Emma? How would you modify this image to show that? Okay. Where's the concentration of hydrogen greater in this system? I just saw you say, oh. Go ahead. Why? Why? Where's the concentration higher? Good. Higher on the outside. Isohumulones make the membrane leaky. In other words, now hydrogen can pass through by simple diffusion. What's that mean? Which way do molecules move in simple diffusion? Yeah, high to low. In other words, if you wanted to model this, these hydrogen ions, where are they going? Working their way back into the cell. Okay? Now use this to help explain what's going on. How is this going to affect the electron transport chain? Chemiosmosis, ATP production, oxygen consumption. All right, go ahead and try to work on that. That, I suppose, I get asked about with tests a lot is, do you want complete sentences? Do you want it, can I draw instead of writing? I say do what's going to be most beneficial to you, but you still have to answer the question. And what I mean by that is, if you're going to draw something like this, if it says model what's happening, you got to tell me what's going on. That you've still got to mention things like, or put labels in like, electrons are moving down the electron transport chain. Hydrogens are being pumped against the concentration gradient to the outside of the cell. They're diffusing back in. Those are kind of things that are labels that I might expect to see that help to bring this model to full fruition. Okay? So know that there are different ways that you can express yourself, I suppose, on the exam if you want. What is this going to do to... Well, there's a couple of things we need to look at. What does this do to the electron transport chain? What does this do to oxygen consumption? What does this do to the hydrogen ion, or the pH, or hydrogen ion concentration across the membrane? And then what does this do to ATP yield? So let's just start with the pH. If we have an isohumulone, normally pH just outside is somewhere around maybe 6.5, maybe a little bit lower than that. Would the pH be higher or lower after the addition of an isohumulone? Higher. Concentration of the hydrogen ions decreases, therefore pH will go up. What does that do to ATP yield? Less. Why?
Because there's less hydrogen ions outside of the cell that can be used for ATP synthase.
Yeah, and the other way that I look at this too, let's imagine this room was full of whatever, I think this seats like close to 60 or so people, okay? And let's say that at 10:59 at the beginning of class we said, all right, everybody, no, let's say 11:49, end of class. I say, all right, time to go. And everybody tries to jam out this door. It's going to take quite a bit of time, but this is like what happens with the ATP synthase. Everybody, all 60 people going through this front door, that generates a lot of energy in the case of the ATP synthase. What if you start to create more doors to allow for those hydrogen ions to go from the outside to the inside? So what if we opened a second door? There's less energy. There's less people that are going to go through this one. And that's exactly what's happening. Is because there's more space or more areas where those hydrogen ions can get through the membrane again, the amount that's fitting through this, it's almost like a funnel in some ways, is going to be greatly reduced. So less ATP as a result of it. What about electron transport? Does this affect electron transport? Who would say yes? Okay. Alexi, why do you say yes?
I'm guessing because the electrons don't need to push the hydrogens across.
Don't think of it as a don't need to. It's
They
it's
don't put as much energy into it.
no. This is a great conversation. I think one of the things that we sometimes try to do is we try to put emotions behind some of these things, like those electrons want to move those hydrogens across. That's not really what happens, right? It's more that those electrons are moving because of some. It's a physical pull of those electrons from one complex to the next. You're absolutely right. We see less hydrogens outside than we normally would. But the effect itself isn't on the pumping of those hydrogens across. Where's the effect? It's keeping them outside the cell. So in this case, what we would actually find is that electron transport would continue to occur as normal. Those electrons would still move. There's no block of the electron transport chain, so they're still going to move from one complex to the next through this process. What does that mean about oxygen consumption? Same or less or more? Electron transport's occurring at the same rate. Same amount of oxygen is going to be consumed. Any questions on either of these two? I think this was time well spent. Okay, so a couple of things before you pack up to head out for today. Two things related to lab. One, don't forget to start your cultures. Everything's sitting out for you. Okay, so I've got a rack of LB tubes, one empty rack for the 8 a.m. lab, one empty rack for the 10 o'clock lab, and your plates are out there too. So just pick a colony, start a new culture. That's one. 2, dichotomous keys due tomorrow. Okay. However format you want it, that's fine by me. Just make sure that I've got it either in Blackboard or a hard copy by lab. There is no lab quiz tomorrow. You should not have to focus on that. And then last, practice. Practice, practice, practice. Okay? The essay question, it's going to be like something we did today. All right? Just to give you a fair heads up. Yes. Yeah, thanks for asking that. There will be, so the way that this one is broken down is there's a dichotomous key problem that is maybe a bit more extensive than just identify. There are 2 short essays that could probably be done in 2 to 3 sentences. And then the one that I'm labeling as an essay, I'm asking you to write a couple of sentences, a description, and then a larger answer. So it's going to be actually quite similar to like the workflow of what we did today. All right. And the last note on practice, you don't learn how to, I don't know, downhill ski or become Shaun White just by sitting on the couch and watching him, right? You actually got to do it. All right. So, with that inspiring Olympic moment, have a great rest of the day. I'll see you tomorrow.
You want me to start a culture?